The practice of science has shaped the modern era. But how are discoveries made? And how does science progress? 3 British scientists, world leaders in their fields, have changed our understanding of our universe, our planet, and ourselves. A physicist whose mysterious radio signals from space rewrote astronomy.
She actually recognized that there was something happening. I suspect that perhaps only 1 in 100 people would have spotted it.
A chemist whose radical theory about our planet divides the scientific world.
He's one of the greatest thinkers of the current age and destined to go down in history.
And a biologist who discovered the secret of life in a sea urchin.
Your fundamental discoveries have profoundly increased our understanding of how the cell cycle is controlled.
Their stories tell us about the nature of scientific inquiry in the modern world, about how scientific breakthroughs are made, and about the workings of the scientific brain.
Scientists are very keen to get at the truth, to get to the bottom of things, but there is no such thing as ultimate truth. Every time, you turn over one stone and you find something interesting underneath it, and then that just leads you to another stone, and there are layers. It's sort of real onion skin. You go down and down and down and down and down, and there are always wrinkles to be figured out. And what's curious, I found, is that some things are very easy to find out once you know how to find them out, and some things, very simple questions, turn out to be amazingly difficult, and it's utterly unpredictable which ones sort of fall out and which ones don't.
Tim Hunt's remarkable career has led to the understanding of one of the greatest scientific mysteries of all: cell division, the complex process in all plants and all animals that underpins life itself. That it happens, and even why it happens, are straightforward propositions. But until Tim Hunt, nobody knew exactly how one cell divided to become 2. The unraveling of that key mechanism was so fundamental to the understanding of life that in 2001, Hunt was awarded the Nobel Prize. It was the crowning glory of a life devoted to scientific research at the very highest level. Recognition of a discovery that owed its existence to Tim Hunt's singular approach to science.
One moment you're ignorant, and the next moment you sort of get that little tiny, tiny clue that sort of sheds an enormous amount of light on the whole thing. There's a sort of mysterious thing about science. It's so different from engineering. I mean, you've been able to build a bridge over a river for thousands of years. The basic principles are understood. But in science, you're never quite sure where you are, actually, most of the time. And you've got to enjoy swimming in this sea of unknowingness. Otherwise, what's the point?
There's a famous quote from a physicist named Leon Lederman which says, "Those who never stop asking silly questions grow up to be scientists." And Tim, to me, is the exemplar of that sort of person, because there is nothing that Tim doesn't want to know the answer to.
One of the things he thinks about is why the sky is blue, and questions like that. And what I think we're seeing is a very curious mind, the sort of mind that a child has, which always asks questions. And most people lose that curiosity, but Tim has it in spades.
I like to tell the story of having learned that I'm a Nobel laureate, of my daughter, who was then 7, Celia, saying, "Daddy, why is the ceiling opaque?" And I looked up at the ceiling. Well, light doesn't get through the ceiling. But then I looked out of our window, and I thought, oh, goodness, how does light get through the window? And I realized we'd done all this other measuring refractive indices for A level, but nobody had ever told us why light gets through the glass but doesn't get through the wall. And this seems like a really sort of rather fundamental thing. So I then started asking people, how does it work? How does it work? How does it work? And physicist friends and biologist friends and absolutely everybody I ran into, why does light get through glass? And people, it was so interesting. So different people use different approaches. This is a good example of how a biologist thinks about things. So when I knew that glass was a frozen liquid. So I thought, well, what other liquids? Maybe it's because it's a liquid that light gets. And then I suddenly realized that mercury was a liquid, and light is reflected off mercury. It doesn't go through mercury at all. So that hypothesis was out the window. And then I thought about carbon, and carbon comes in the blackest black that absorbs light better than absolutely anything on Earth, but also in diamond, which is the sort of sparkliest, most translucent stuff you could possibly imagine. And that then tells you that it's to do with the electrons, how the electronic configurations of the actual material. And then you realize that it's to do with the theories of how photons interact with electrons, and then you're lost, basically.
He writes a problems book with his friend John Wilson, and they once worked out how far up an Alp one Mars bar would take you. It's that sort of thing. How many calories are there? What do calories translate to in terms of vertical motion for a human? So he's doing that, I think, in his head most of the time, except probably when he's looking vacant, when he really is vacant. He must have some of those moments. But I think he is continuously thinking about most of the situations he's in and wondering what's happening. Yeah.
Tim Hunt's passion for scientific inquiry started when he was a small boy.
I just loved old radio sets, and there were a lot around. They were just so amazing and so beautifully made, and they were the tuning device. I guess I longed, actually, to be an electrical engineer and be able to sort of design the circuits, but I was completely hopeless. I couldn't do it. I loved it. I loved making motors and microphones and loudspeakers. Always wanted to understand how things worked. Here you stick this Bakelite box on the table and you tune the dial, and, voices come from the universe towards you. And then you look inside, what's doing this? It's just these glowing valves. I remember making fuses too. That was good fun. So you soak a piece of string in sodium chlorate, and then I remember one time we tried to accelerate its drying out, so we put it under the grill, which was a big mistake because the thing went kaboom.
He was older than I was, so he was the leader and I was the follower behind, if I am to be absolutely honest, which was good. He was good. He was imaginative about it. We did interesting and fun things. I suppose we were quite scientific about it, really. Now I come to think of it. I think he's very sort of inquisitive. He likes to try everything out.
I just wanted to know how things work all the time. All that, how do things... what's behind this, and I loved the idea that you could, there was a sort of simple explanation behind everything you could see.
This passion to really understand how things worked persisted and shaped his entire scientific career. It led him to the challenge of understanding life itself and to the fundamental process that drives it.
Our lives start with a single cell. That first cell splits to become two, and then they in turn divide into two to become four, and so on. So after just 48 doublings, you have 100,000 billion cells in your body, and you're ready to spring forth as a human being.
The miracle of cell division is, at its most dramatic, a process that creates new life. But it's also fundamental to maintaining life, and it's this that reveals the complexity at its core.
Everything is being replaced all of the time. And there are new molecules being put in place of old ones. The old ones are sort of recycled. And I always remember when I went as a junior proctor, we went and visited this Air Force base. And I was very thrilled because we saw a Canberra bomber So I asked, gosh, is this really an original? And they said, well, the pilot's seat might be the original thing, but everything else got new wings, it's got new fuselage, it's got new this, new that. The thing had been totally replaced. And yet it was still a Canberra bomber and not some other kind. It wasn't a Spitfire, it wasn't anything. So we're like that. Life and cells have the ability to patch themselves all of the time. Damage is being done all of the time. You have a new nose, basically, certainly every 7 years. I mean, there isn't a single component in your nose left. But the amazing thing is that the nose still is your nose. It hasn't grown, it hasn't shrunk, it's still got the same shape that you were born with, so to speak. Absolutely remarkable, So how these cells know? I mean, there's a cell here in the tip of my nose that knows it's a cell in the tip of my nose, and if something happens so that cell dies, then, its neighbours say, oh dear, Tim's lost the tip of his nose. We'd better put in a new cell there because it's gone, and that's replaced by cell division.
For Hunt, understanding this great mystery means breaking it down to its smallest components. Life, all life, can be understood as a series of complex mechanisms, a myriad of individual processes happening within the cell.
The way I think of it is that, you imagine yourself shrunk down to the size of a molecule, and you're standing by this amazing molecular machine and watching how all the things, come in and out and pull each other and push each other. And those are the terms in which you want to understand it.
There are different philosophies of how you should do science or can do science. And one philosophy is reductionism, the idea that you can explain everything in terms of the smallest bits inside it. And like explaining the whole of nature in terms of atoms or quarks or subatomic particles. And that way of thinking has led to molecular biology. The way to understand life is to understand the smallest things in living organisms, molecules, DNA, proteins, and so forth. And that somehow, out of understanding all the details, the big picture will emerge. Tim is definitely working within that tradition.
This approach had started when he was a teenager, when he came across a problem that would take him more than 20 years to solve.
When I was doing A-level biology, I remember seeing an amazing movie of biochemical pathways with compounds flowing down and cycles going round and things spinning off and all that kind of stuff. And I remember thinking at the time it was all very well, these pathways, but there didn't seem to be any control anywhere. So how could you have all those reactions going on simultaneously? There's got to be something controlling it. This was very thrilling, a kind of revelation. So what I suddenly realized was that control was the fundamental issue of all life. From then on, I knew that being a biologist was what I was cut out for.
Tim Hunt was brought up in Oxford, where the disciplines of the academic life and a passion for detail were part of his heritage.
Dad was something called a keeper of the Western manuscripts at the Bodleian Library. He had a sort of encyclopedic feel for what was where, and you could use these manuscripts, which are actually bound into books, to trace who knew what when. Manuscripts were copied by monks. There was no printing in those days, so everything had to be copied. And the monk would make the odd mistake. But those mistakes then tended to be copied faithfully by the next copyist, and then that book would be taken to the next monastery. And so you could actually trace a genealogical tree of the spread of knowledge across Europe by this technique. His office at the Bodleian was quite remarkable. It was a huge big table, quite a big room, and in the middle of the table the papers, the letters, were about a foot deep or something, and then towards the edge they sort of thinned out rather, because otherwise they'd have spilt onto the floor. So I don't know how he managed his correspondence, but it's exactly, I'm exactly the same. It's a sort of classification system whereby the less important things fractionate to the bottom of the pile, and the important things either stay on the top or rise to the top.
Tim and my dad are very alike in many, many ways. They are both very scholarly. Tim is a stickler for science and the way that it is carried out, and for all the principles of keeping science honest. My father was an absolute stickler for scholarship.
Dad was very keen on the right way of doing things, and I've always felt that too in science. And that served me well, because it's the details in biology that unlocks the secrets.
Hunt's academic career began in earnest when he went to Cambridge to read natural sciences. He found himself thrown into a world of scientific optimism and mild eccentricity.
Tim and I were very close friends. We used to do a lot of things together. We were often called the Terrible Two. I'm not quite sure why. I don't think we were particularly terrible, but they were... I think people found us a sort of unusual pair of people to have around. Most people had just ordinary white lab coats, but Tim and I dyed ours, and his most characteristic clothing at the time was a pink boiler suit that he used to wear at the lab and indeed walking around Cambridge. And in the winter, this was, it wasn't warm enough, so he needed something warmer. Then he had sort of third-hand fur coats. So he wore these rather eccentric, long fur coats over his pink boiler suit. You never felt that it was an affectation in his case. He made eccentricity seem normal. We found rooms in the Eagle pub, and we lived there for 3 years while we were doing our PhDs. We were nocturnal, so we used to go back into the lab and start work at about 10, and usually work through till 3 or 4 in the morning. And we had loudspeakers in both our labs, and we draped wires over the outside of the building. And so at night we had the whole laboratory resounding to this wonderful music, sort of Monteverdi operas, symphonies, going on all night in the lab, and the whole lab echoed. So we had a very nice way of working there.
Hunt had arrived at Cambridge at precisely the right time for a young scientist who was passionate about the emerging science of molecular biology.
I think Cambridge was a good place for high objectives. It was a site of great science, and you were supposed to do it in a sort of understated way. And when I think back at it, it was probably then, in biology, the leading university in the world, if you just put what people were trying to do. There was no other place which had the focus on important things.
Dr. James Watson, aged only 34, an American biologist who worked for 2 years at Cambridge. Francis Crick, aged 46, worked in the Admiralty on mine detection during the war, then switched to biology. They puzzled out the mechanism of inheritance that makes you so much like your parents. Between them, they shared the Nobel Prize for medicine.
I think Tim was very impressed by the molecular approach that Crick was advocating, and indeed most people were advocating, and that was the sort of high noon of molecular biology. The genetic code had just been cracked, the mechanism of protein synthesis had just been worked out. Tim was working out some of the details of all this. It was a period of enormous optimism, and people thought that this was the way forward to understand life.
When Watson and Crick lounged around chatting about things about which they knew nothing, but as a result of this, they discovered the structure of DNA, the most important, arguably the most important scientific biological discovery of the 20th century. There's more than one way of doing good science, and that's extremely true. You know, I mean, you just have to take it's so hard. I think that's what a lot of people don't understand. It's so hard and so slow that you have to take little clues from wherever they come from. Jim Watson was a fellow of my college, and don't quite know how I first met him, but I mean, he sort of would just drop by. Jim has an amazing property. He sort of drops by. He sort of pops out in your life at crucial moments, and it's a very interesting man. Very interesting man. You could actually see that he also really, really cared about getting to the bottom of things, and that's what's finally important. I mean, it's people who are actually trying to get to the bottom of things, not for their own glory, but just because they really, really want to understand. It's terribly important.
Cambridge was always rather amusing at those days, because Francis would have about one idea, I think, every 2 weeks, and it'd take a couple days for him to be convinced it was no good, and in those couple days he'd never stop talking, and if we were in sort of good spirits, we could, really ask him difficult questions.
I'm very much a hero worshipper. Francis Crick was really our hero. He was an omnipresent figure at seminars. He'd often be sort of sat at the back of a seminar, and he would almost always ask questions, sometimes if it was okay, within the body of the talk. And these questions were always trying to just clarify things, to get to the bottom of things. And unlike some people who asked in Cambridge, who asked questions to show how clever they were, actually they were often questions which revealed how ignorant he was. But he still had the confidence to ask, because if you didn't understand that particular point, the whole thing just would be incomprehensible.
Certain things, I think, can be thought to be alive. I think we would agree that you and I are alive. The difficulty comes with the borderline cases, like the viruses. And I think really what it comes to is you're really arguing about a word when you ask the question. Is the polio virus alive, for example?
Tim, because he started working on how proteins are made, the process called translation, he was very connected to those people, because that was part of the central dogma. DNA makes RNA makes protein. And the question is, how do you make protein? What are the nuts and bolts of those steps? And how do you control that process? And that's really where Tim started as a scientist.
It was the beginning of a journey that would lead to Hunt unraveling the fundamental mystery of cell division. A journey that began with understanding the biochemistry of proteins.
Proteins do absolutely everything. I mean, I used to enjoy telling students when we sort of started out on a journey through cell biology and biochemistry that, when I look at your face and I'm looking at proteins, that's your skin, and I can see that your blood is red because your lips are pink, and the eyes are clear, but that's protein specially arranged so that the light can go through it. So the hair is a different kind of protein from the skin, and related but different, and so on. So that's all protein. Everything you're made of, stuff you can really sort of feel. I mean, it's tough stuff, and if you've ever eaten a piece of gristle, you can sort of know how ghastly and tough protein can be. But they can also catalyze chemical reactions to make sure that what you eat then becomes you, and you alone, because you break down all the proteins in their components and then build them up again into your own cell's components.
At Cambridge, Tim Hunt began working on the problem of how individual proteins were made, and specifically how this process was controlled.
What became clear, and what was nice about all of this stuff, is that you could turn protein synthesis on and off. And it was the fact that you could turn it on and off meant that you were really studying the control of protein synthesis in a test tube. And finding out what the molecular mechanisms behind that switch was became a sort of all-consuming goal.
In 1968, Tim Hunt received an invitation that would change his life.
I'd come to New York to work with a man called Irving London, who was actually the head of the Department of Medicine at the Albert Einstein College of Medicine. But he was very keen on research, and he was interested in exactly the same problem as I was, which was the control of protein synthesis. I lived on the Upper West Side, not far from Columbia University, really, but had to travel to the Far East Bronx. I mean, it's probably about a 40-minute subway ride. through some of the most roughest, crime-ridden districts in the world, so there was no question of looking respectable.
This is 125th Street.
But I used to read the New York Times from cover to cover going out, and the Journal of Molecular Biology coming home, quite often, usually pretty late at night.
It was a cultural as well as a scientific opportunity for Hunt, who went about documenting his time in the US by taking photographs.
I saved up enough money to buy a Pentax camera that had actually a little meter built into it. And this was sort of the beginnings of the real revolution, so that you didn't have to worry about exposures and all that kind of stuff. Very obsessed with these sort of great French photographers who could somehow sort of capture the minute of the passing scene. I absolutely loved that.
In the lab, Hunt and Irving London were trying to capture significant moments in protein synthesis. They separated the individual proteins manufactured in cells using chromatography. Each horizontal line represents an individual protein, and each column a different point in time.
You have to keep on taking time points, and, I don't know, half a dozen, maybe 10 samples per time course would be enough.
The technique made it possible to see the ebb and flow of proteins as they were produced in the cell, in a way that taking a single measurement could not.
I learned a great deal about technique, measuring the rates of protein synthesis and being precise about controls and making sure that you saw the big picture and not, it's sort of like making a movie rather than just taking snapshots.
In New York, Hunt had equipped himself with the tools he'd later need to make his greatest breakthrough. But as is so often the way in science, serendipity and chance encounters also played their part.
About this time I went to a talk by a guy called John Gerhardt, and very soon I was right on the edge of my seat because he was explaining his studies on MPF, maturation promoting factor, the magical missing link which seemed to be the trigger for cell division.
For years, biologists had known that, theoretically, something must make a cell divide. But try as they might, they couldn't find it. They had, however, given it a name.
I think there's an interesting lesson to be learnt from MPF, because when it was originally conceived, it had a rather abstract nature. It was just a factor that was present in cells that made other cells divide. But one had no idea quite what it was. And what you needed to do was to turn that abstraction into a physical reality. And you do that by determining what molecules make it up. And science quite often inhabits that territory. You have an idea, you have an abstract thought about what's going on, but you have to turn it into something more concrete.
It was nowhere near what I was working on at the time, but it struck me as a really delicious problem. You could almost taste it within grasp. You could begin to think about what it was and how it might work.
Hunt's interest in the stubbornly elusive MPF stayed with him. A decade later, he was dividing his time between Cambridge and the US, looking at protein synthesis in cell division.
Woods Hole is a famous marine biological laboratory at the seaside near Boston, and that was in the summer of 1982, alongside a friend, Eric Rosenthal, who was doing some experiments on clam eggs. And what we were doing was to try to compare the kinds of proteins made in clam eggs before and after fertilization.
With Tim's help, Eric showed that the proteins that you make before you fertilize a clam egg and afterwards are different. And there are 3 proteins that you make much more of after you fertilize the egg. And they got the imaginative names A, B, and C. And so when Eric would do experiments, in each experiment, he always saw a lot of C. And in some experiments, he saw a lot of A and B, and some experiments he saw almost none.
So my friend had been taking snapshots comparing the patterns of protein synthesis, and he noticed that sometimes the proteins were there and sometimes that they weren't. It was just a complete mystery to me. Why would the proteins sometimes be there and sometimes not be? Normally proteins were just synthesized and then stayed there. They didn't just go away. I'd never seen anything like it before.
It was an intriguing problem, a problem whose solution might provide another piece of the MPF jigsaw, and one that Hunt was ideally placed to investigate.
What you really needed was a movie to understand what was going on, very much the same kind of approach that I'd developed in New York.
Eric never had the time or the inclination or, the desire to overcome the experimental difficulties to sort of take that leap and pursue that. And Tim saw something that he didn't expect to see. And then in that situation, scientists have to make a decision. Do I stay narrowly focused on what I thought I was doing and just ignore this thing as being a will-o'-the-wisp? Or do I realize that I've seen something that is much more important and pursue that?
I started my own research on sea urchin eggs to see if the same kind of things were happening. I just wanted to get to the bottom of it. The experiment was very, very simple. That's the important thing. So one day, and it turns out the experiment started at 8 o'clock at night, and the others had all gone out dancing or drinking or something. So I was left all alone in the lab with just me and some sea urchin eggs. And I checked they were fertilized, added the label, and then took out samples roughly every 10 minutes. We just analyzed the samples on something called an SDS polyacrylamide gel, which was very standard and we knew exactly how to do, and do it very beautifully, though I say it myself. I saw this protein, which was actually the very first one whose synthesis you could just detect. And it got stronger and stronger and stronger, and then it faded away. All the other ones just went on getting stronger and stronger. So I looked at this, and this is very extraordinary. And the way the experiment was done, there was no question but this protein going away must mean that it was being degraded, and being degraded specifically. I mean, there was just this one protein went away. All the others behaved as you would have expected. Looking at the full picture in this way, taking samples over time, let me see that the protein wasn't coming and going randomly. It was coming and going in a very distinct cycle. I looked at this and, gosh, that's absolutely amazing. There it was, on this gel in black and white.
Hunt called the protein cyclin because it came and went with the cell's cycle. He knew that what he was witnessing was significant, but he had no idea what it could mean.
As it so happened, I think I must have gotten the result on a Friday morning. On Friday evening, I ran into John Gerhardt at the wine and cheese party after the Friday evening lecture. John was the man whose talk about MPF, the magical missing link, had excited me so much all those years ago. So suddenly, everything was coming full circle. This was the most exciting conversation, scientific conversation of my entire life. I mean, bar none. It was extraordinary. I remember sort of somebody came up and tried to talk to me. I just brushed them off because this conversation I was having was so important, to get it right. And it was absolutely the key. And so from that moment on, this is very unusual. The other things I've told you about, it often took weeks or months to figure out what was going on. But basically, I'd seen this protein go away, and I knew in my heart of hearts that somehow this was really important and somehow deeply connected, intimately connected with the cell division control machinery. And it was something that nobody had ever in their wildest imaginings thought of, that the protein might actually just go away.
At the time that Tim discovered cyclin, it was sort of like the Northwest Passage. It's like knowing that the Northwest Passage exists, but I can't point to it on a map and tell you exactly where it is. And so the search, if you like, is for that discovery of what is the molecule that is MPF. And so when Tim discovered cyclin, there's immediately this congruence between, here's cyclin, a protein we have to make to get in cells to divide, And here's MPF, something that pushes cells into the stage where they're just about to divide.
Why did I have this sense that the going away of cyclin was intimately related to the control of the cell cycle? And why did that explain so much that I internally felt intuitively that it was probably the right answer, although it had to be, you know, you had to nail it. You couldn't just take it for granted. And I'm not sure that I can give a very coherent, you know, because it was more of a gut feeling. The truth of the matter is I didn't dare hope that my cyclin, which was going up and down like a yo-yo, was part of MPF. I mean, that would have just been way too good to be true. I mean, that would have been sort of like an instant Nobel Prize. And, you know, you don't get instant Nobel Prizes just by doing one dumbass experiment on a Saturday morning, so to speak, you know.
The excitement felt in Tim's circle at Woods Hole was not matched in the wider scientific community, and Hunt was brought abruptly down to earth.
People looked at me as though I was slightly deranged. I had quite a hard time. Indeed, when I sent off our first paper about this work to the journal Cell, the editor wrote back to me, "Dear Tim, the good news is we will publish your paper, but the bad news is in nothing like its present form." And one of the reviewers said it was wild speculation based on faulty logic.
I can remember him ringing up and saying that he was, I can't remember what phrase he used, but he was essentially saying that he was tired of science and of research.
I think most people didn't have any problem with the fact that it seemed to have something to do with the cell cycle, but showing that it controlled the cell cycle was a much trickier thing to actually establish.
He is a stickler for the world of people, peer recognition and all the rest of it. Well, his peers didn't recognize him, did they?
They were just sceptical, and rightly so in a way. That, you know, let's reserve judgment, put it that way, right? I think that's a perfectly legitimate thing to do. We had a lot of work to do, and it was, I mean, this was one of the funnest periods of my life. A little bit scary, but good because people were so skeptical, nobody was still prepared to make that plunge themselves, right? And that was kind of, so it gave us a lot of space, actually.
Tim is not one of those arrogant scientists. He actually suffers a lot from self-doubt. Often people are black and white, you know, this is right, this is wrong. Tim understands greyness. He understands that sometimes problems are not very clear, and the best thing to do is talk about them. And these are features that mark out, in my view at least, a very good scientist.
Hunt was faced with the daunting task of explaining exactly why the mercurial cyclin was in fact MPF, the stuff of life. His first instinct was to collaborate.
Once you've got an idea, it's very good to bounce it off other people. You know, you've got a result and you think it means this, and you find your peers, I mean, that is to say your competition, the people who are actually working on this, you know, and trying to sort of tease out as much as you possibly can to sort of suck the data absolutely dry, you know, right down to the bone, so that you can, you know, so I very interesting, the sort of the relationships with competitors, and you know, I always found the best thing was to tell everybody everything.
It was Hunt's spirit of openness that led to his fruitful collaboration with the man who would be instrumental in helping him complete the cyclin story.
I had been working on sort of genetic approaches to how the cell division cycle is regulated. I've been doing that since the sort of early to mid-1970s. And my work was abstract, and I didn't have any idea about how the biochemistry, the chemical molecules that were involved. But I was very interested in that. And in the early 80s, I was gradually getting towards looking at what molecules might be involved using genetic approaches. And so I started to get very interested in other people's work which might illuminate that. And I also was going to conferences and meetings where Tim was talking, and he was talking about this protein that was appearing and disappearing. So I obviously thought this could be, if luck was in the right place, this could be an element of the same sort of problem that I was interested in.
If cyclin was to unlock the mystery of the magical MPF, it would be necessary to explain its precise role in cell division. What's more, it would need to be present in the dividing cells of all living things, not just a single species. Tim Hunt, Paul Nurse and their colleagues set to work in earnest.
We all knew each other, and we all worked on different things. You know, she worked on clams, he worked on yeast, I worked on frogs and sea urchins, and you know, it was great, and there was so much to be found out. The first thing, you see this in sea urchin eggs, and you say, well, okay, that's very interesting, so that's how sea urchins do it. Then you see it in clams, and that's pretty significant because actually clams and sea urchins, if you look at the tree of life, they're pretty far apart. I mean, so that suggests that it's, at least in all sea creatures, mollusks in echinoderms. Now, we're descended from echinoderms, so if echinoderms do it, probably we do too. To go then from frogs and to try and make the MPF connection was really important. So once we knew it was there in frogs, we could be utterly confident that it was going to be there in humans. And then it was clear this was absolutely universal.
This time, there could be no doubting the veracity or the importance of their discovery.
Every single animal, every single plant uses exactly the same fundamental mechanism to catalyze cell division. And by the time we knew all this, it became pretty much irrefutable. Suddenly people, you know, the scales fell from everybody's eyes and everybody realized that this was the secret, this was the basis of cell division.
One of the greatest mysteries in biology had been solved. The continued existence of all life on Earth could now be explained. Cell division was no longer governed by theoretical construct, but by real chemicals on a real protein called cyclin, discovered in a sea urchin by Tim Hunt. Now, the scientific establishment beat a path to his door.
The Nobel Prize is the only scientific prize that the public generally know about, so it is iconic. I mean, there are quite a few other prizes which sort of lead up to it, but it's the Nobel Prize that really matters.
Tim phoned me and he had to come down to London from his lab for a press conference with Paul Nurse. So he dropped by my office and he looked like he'd been in a road accident. He was in true shock, I would say, very pale, very shaky. And we went to tell Celia at that point, who was at primary school and sort of old enough to understand. And the head teacher said that when we got to the door, we looked like we'd been in an accident, basically. And we were going, won the Nobel Prize, what shall we do?
And Tim really didn't believe it. He almost thought, I think, that it was a spoof, although I think he had talked directly to the Swedes, whereas I had not. And I said, I don't think people would be so cruel, Tim, certainly not to you.
Until the king had actually given me this bloody medal, I really thought that somebody might come in and no, sorry, made a ghastly mistake.
Dr. Hartwell, Dr. Hunt, and Dr. Nurse, your fundamental discoveries have profoundly increased our understanding of how the cell cycle is controlled. This new knowledge has a huge impact in cell biology, with broad applications in many fields of biology and medicine.
It's very moving, because you know this is the pinnacle of your scientific career.
And I now ask you to step forward to receive the Nobel Prize from the hands of His Majesty the King.
Oh yeah, I still feel enormously proud. Hugely so.
I think the Nobel Prize rewards significant achievements in science, which are of importance for science as a whole. And the Nobel Prize is really about rewarding things which become foundational parts of conventional science. The key discoveries that are the foundations of established science. And discovering the way that cell division works is a key discovery in history, in the history of biology. And I think that's why Tim's Nobel Prize was a Nobel Prize.
There are always these pioneers who work things out and who had the idea first. And I think it's absolutely fine to honor those people, actually. I mean, if you look at the list of biological Nobel Prize winners, it really is a sort of, a roll call of major discoveries down over the last 100-odd years. The question that I always ask when I'm trying to understand science, well, gosh, why do they know that and they don't know this? And then if you go back to the history, you say, well, we know this because we were working on that and we accidentally found that, and then that led to, there is a sort of thread running through it, kind of continuity of ideas.
If you like, it's all just this gradual opening of a flower, an intellectual flower, that takes you from the golden age of molecular biology through worrying about how you make proteins to discovering the protein that controls cell division. I think one of the things that's so fantastic about, all the fame and, publicity that accompanies things like getting a Nobel Prize is that lots of the people who go there, nice or not, are people who are just unbelievably ambitious. And Tim, certainly, like most scientists, is competitive. He wants to do things that are important. But he's really a scientist in a way that I think we've a little bit lost, the sort of almost 19th century tradition of the independently wealthy gentleman who does things just because it's fun. And I think Tim's spirit of fun and generosity and excitement about science, as little contaminated by personal ambition as it is in him, makes him such a remarkable person.
I have here his Nobel medal, which he kindly gave to the college because he thought it would be locked up in a bank, but if it's in Clare College, at least it would be seen. He's that sort of man, very generous. And we miss him here because when he moved on to greater things, of course, we lost him, but it was always life and soul of the party, always good for a laugh. Cheered us all up. In fact, still miss him. I love to see him when he comes here. He still comes on a regular basis, but not enough for me.
Today, Tim Hunt works for Cancer Research UK. It was a move that had a special poignancy for him. Before he was awarded his Nobel Prize, his mother had been diagnosed with the disease.
I got this postcard from her and said, had lovely holiday in Scotland, but ate poisonous mushroom. And it wasn't the poisonous mushroom, it was the fact that her liver was already sort of just one horrible mass of it had stopped working, basically, and that's what was upsetting her. And it took a little while before she was very funny about it. I mean, they took her down to some X-ray machine, and the young intern sort of took one look at her X-ray and said, "Oh my God!" And she trained as a physiotherapist. She sort of knew what hospital life was like, and it amused her very much that, he behaved so badly. I mean, it's absolutely not what you do in front of a patient. But she was funny like that about, very funny about herself. The cancer had by this time turned into something called ascites, which means just sort of free-living cancer cells in her belly. And I tried to read, I didn't know nothing about cancer at that point. I said, how can it be that these cancer cells are sort of actually sucking the goodness out of her good tissues? And why is it the muscles waste away? And her brain was as sharp as anything, right to the end. I mean, there's no sign of mental deterioration whatsoever. It's just her body was failing. So, I mean, as a scientist, that really fascinated me. And I longed, and I have ever since that day, longed to work on that. It was announced in October or something like that, and she was dead just before Christmas. One time she came into the room and said, "I want you to know I'm not afraid of dying." After I'd heard that I'd won the Nobel Prize, it was a very, you sort of, it's hard to describe the emotions that go through your mind, but one of them was I was sort of just driving out along, just coming in, and, oh God, what it's called, along the Finchley Road, basically, the A41, and suddenly sort of tears welled up in my eyes because I thought how proud my parents would have been, and it was really so awful, actually. Weird. Just weird. I was very thrilled when the opportunity came to work in a cancer institute, partly because of my mum, and partly because I think it's just a very, very interesting problem, and partly because an awful lot of people die of it. Understanding how the cell cycle works does kind of inform aspects of cancer research, but and as I often say, if I did discover a cure for even one cancer, nobody would be more thrilled than me, but it's not what motivates me, for one. I just want to try and understand what the hell is going on.
Cancer is characterized by cells dividing out of control, but understanding why they do is a problem that has resisted solution for over 50 years.
The time has come in America to turn toward conquering this dread disease.
It is perhaps the last great battle that modern medicine has to wage.
The time has come in America when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease.
But it's not just an issue of funding and political will.
I think that we are still remarkably poor at dealing with cancer. And so when I teach students about it, I draw the analogy between John Kennedy declaring that Americans will put people on the moon, and I say exactly how long it took and how much money it cost, which is a lot. And then Richard Nixon declaring war on cancer. And so he set out as a public aim defeating cancer. And we've had more than 30, probably approaching 40 years since then. And when I talk about that, I say putting people on the moon required 19th century, or actually even earlier physics, right? Basically Newtonian physics. It's an engineering challenge, it's not a scientific challenge. The problem with cancer is it's a scientific challenge, right? We don't understand exactly what cancer is. Knowing the molecules that control cell division helps us a little.
If anyone can make headway in the defeat of cancer, it's Tim Hunt. Not because he is better funded or has a superior work ethic than his fellow scientists, but simply because of his undiminished thirst for discovering how things work.
Quite often it happens that problems get solved out of left field by people just coming at it a different way. It very, very rarely happens that biological problems are solved by a direct frontal attack.
Tim Hunt's career is in many ways typical of the 21st century scientist. A rarefied world of research institutes and searching peer reviews, of academic conferences and sparking ideas off fellow scientists. But there is a particular cast of mind that can go beyond even the most cutting-edge science.
There are two sorts of scientists. There are people who are really interested in big ideas, and experiments to them are simply a mean to big ideas. They're the big thinkers, the speculators, experiments, unpleasant means to an end, often. And then there's another class of scientists who just like to fiddle, to tinker. And to many of those people, speculation is anathema. It means going beyond your data, in a sense, going somewhere where almost emotionally you're not supposed to go. And what to me is unique about Tim is he's both of those people at once.
I think my main talent is sort of spotting sort of funny little things that don't quite make sense, and that, you know, Isaac Newton talked about, you know, shiny pebbles on the beach, like a little. He said I was like a little boy, you know, wandering around, looking for shiny pebbles on the beach. I'm quite good at looking for shiny pebbles and seeing things which, you know, aren't actually on the mainstream, but which lead you off in a new direction and are incredibly illuminating about what's going on. I mean, in other words, I'm a good discoverer of things. Discovering things is the most fun, I think, you know, because they tend to sort of illuminate whole fields in a way that you couldn't believe. You know, one moment you're ignorant, and the next moment you sort of get that little tiny clue that sort of sheds an enormous amount of light on the whole thing. It's not up to me, I don't think, to decide whether my mind is beautiful or not. But it's my mind, and it's taken me to some exciting places and answered some very interesting questions, and it's given me a lot of fun, and for that I'm very grateful.
And you can find out more about the Year of Science on the BBC and sign up for our World of Wonder newsletter at the BBC's Science website. Next tonight, stay tuned for comedy with It's Only a Theory.